Hey, good afternoon. So when we think about protein structure, we usually stop at four levels. But inside real cells, there's actually a fifth level that changes everything. So you already know the basics. Primary structure is the amino acid sequence. Secondary structure breaks them down to alpha helices and beta sheets. Tertiary structure is the 3D fold or the native state for protein. And quaternary structure is when we have multiple units assembling, such as an insulin or hemoglobin. We've learned that in biochemistry. So these levels are mostly defined under dilute ideal conditions, such as in test tubes. Yet cells are actually nothing like that. Inside a cell, proteins exist in a highly crowded environment packed with macromolecules. This leads to countless weak and transient interactions between proteins, nucleic acids, and metabolites. This is what defines quinary structure. And quinary structure is a dynamic, non-permanent network of interactions that's shaped by molecular crowding. These interactions aren't strong enough to form stable complexes, but they can still influence protein stability, folding, and function. Quinary interactions can subtly alter the enzyme activity, shift conformations, and even regulate pathways without ever forming a fixed structure. So proteins aren't just shaped by their sequence or binding partners, they're actually shaped by their environment. And that environment adds a whole new level. And that's really what quinary structure is all about. So quinary structure is easiest to understand through systems where weak, transient, and environmentally dependent interactions measurably alter protein behavior inside cells. These are not stable complexes, such as the case for the quaternary structure of hemoglobin or insulin, but context-dependent interaction networks driven by crowding, proximity, and low affinity binding. So here are several well-characterized and biologically relevant examples. The first one is metabolic enzyme clustering. We also call these metabolons. And so a good example of this is in glycolysis with the enzymes of glycolysis, or in the citric acid cycle with oxidative phosphorylation. And so in the case of glycolysis, we have hexokinase, phosphofructokinase, and pyruvate kinase. And these enzymes often form transient and spatially localized clusters rather than freely diffusing independently. These associations are fairly weak and dynamic, and they're not fixed quaternary complexes. The result is that we get substrate channeling where intermediates are passed efficiently between these enzymes. So this organization actually depends heavily on macromolecular crowding and local concentration. And that's the main hallmark of quinary structure. Number two, let's look at lactate dehydrogenase in the cytosol. And lactate dehydrogenase is one of the enzymes involved in fermentation in muscle cells and things like cheeses and that sort of thing. So in-cell NMR studies show that the enzyme lactate dehydrogenase experiences reduced mobility due to numerous weak interactions with surrounding macromolecules. And these interactions slightly alter the enzyme kinetics and conformational sampling. Importantly, lactate dehydrogenase is not forming a stable complex, it's actually engaging in constant low affinity contacts. Next, let's look at green fluorescent protein, or GFP. And this is often used as a probe for studying quinary effects in proteins. And so green fluorescent protein is a 238 amino acid protein from the jellyfish Aquaria victoria, and it emits a bright green light around 508 nanometers when exposed to blue or ultraviolet light. And so it's a vital tool in cell biology and biotechnology, and it functions as a fairly good, non-toxic, real-time marker for gene expression, protein tracking, and bioimaging without requiring any additional cofactors. So GFP is often used experimentally because it lacks its strong binding partners. So in living cells, GFP diffusion is much slower than predicted from a dilute solution. And this slowdown arises from nonspecific transient interactions with surrounding biomolecules. This demonstrates that even inert proteins are influenced by quinary structure. And let's also look at the ribosome, where proteins are actually synthesized. And if we look at ribosome proximal interactions, newly synthesized polypeptides interact transiently with nearby macromolecules, and this includes chaperones and other ribosomes. These interactions actually influence co-translational folding pathways, and the local environment, dense with RNA and protein, creates quinary constraints on folding trajectories. And of course, let's look at also alpha-synuclein in neurons. Now, what is alpha-synuclein? Alpha-synuclein is a 140 amino acid protein that's primarily found in presynaptic neural terminals. And this comprises about 1% of total cytosolic protein in brain cells. And so this is crucial for regulating synaptic vesicle trafficking and neurotransmitter release. And misfolded alpha-synuclein aggregates into toxic Lewy bodies, and this causes neurodegeneration in disorders known as synucleinopathies, and that's most notably in Parkinson's or Alzheimer's disease. Alpha-synuclein undergoes fairly weak transient interactions with membranes and other proteins. So cellular crowding promotes conformational shifts and aggregation propensity. These quinary interactions are then thought to influence the pathology of Parkinson's disease. And then we have phase separating proteins. We also are biomolecular condensates. So this is seen in stress granules and P bodies. It involves weak multivariant or multivalent interactions, often involving intrinsically disordered regions. This leads to liquid-liquid phase separation. They're not rigid structures, but they're rather dynamic assemblies that are governed by their quinary interactions. And so they regulate everything from RNA metabolism to signaling, intracellular signaling, and stress responses. So for example, the MAP kinase signal transduction pathway with ASK1, MKK, and JNK3, or actually 3 ASK forms of JNK would be part of this process. So overall, in summary, quinary structure is not about what binds tightly, but about what's nearby, how crowded the environment is, and how often weak interactions occur. It affects shift equilibrium, altered kinetics, and it influences folding and function. So it's very important for protein structure, and in short, quinary structure reflects the reality that proteins behave differently inside cells than in dilute solution, because they're actually embedded into a dense, interactive molecular ecosystem. So thanks for watching, and I hope you enjoyed this video. Please go ahead and hit the like button and subscribe to the channel and hit the little bell icon if you'd like to stay updated whenever I post new content. If you have any questions, leave them down in the comments below, and I will see you in the next video. Thank you.